Okay, so unfortunately, our very first session speaker, Marcel Sandu, could not be here with us today or virtually. So we're actually going to move on in the schedule to our next speaker, Chinmay Tikhe from Johns Hopkins University. Welcome.
Thank you. Right, good morning, everyone. My name is Chinmay, and I'm a postdoctoral fellow here in the George Dimopoulos Lab in Bloomberg School of Public Health. And today I'm going to talk about a new bacterial-based bioinsecticide that our lab is developing for the control of malaria vector mosquitoes. Now, as we all know, even today, malaria remains the most devastating vector-borne disease globally, and most of the cases of malaria are reported from the African continent. So vector control remains the main malaria interventions. It was reported that between year 2000 and 2015, 70% cases of malaria were averted because of effective vector control strategies. Now, most of the insecticides that are used for vector control are chemical or synthetic insecticides. And even though these insecticides are efficient, there are some There are some problems with the current insecticide treatment strategies. So we have very limited number of insecticides that are available for vector control. Just to give you an example, only pyrethroid-based insecticides are currently approved for the use with insecticide-treated bed nets. And because of this limited number of insecticides, we have a lot of increased number of insecticide-resistant mosquitoes. If you see this graph, it shows the number of countries that have reported confirmed cases of pyrethroid-resistant mosquitoes. And other than the insecticide resistance, there is also a report of behavioral resistance in mosquitoes. So mosquitoes have started biting outdoors. Insecticide bed nets also have very limited lifespan. And other than this, the current chemical insecticides can also have off-target effects on beneficial insects and aquatic life. So this is where our lab comes in. We have been working with this bacteria known as Chromobacterium for the past few years. Chromobacterium is a common soil-dwelling bacteria. This particular strain was isolated from Aedes aegypti mosquitoes in Panama. And over the past few years, our lab has shown that Chromobacterium can have mosquitocidal activity against adult and larval stages of different mosquito species. The most interesting thing about Chromobacterium is that even a non-live preparation of this Chromobacterium is effective in killing mosquito species. So we wanted to find a delivery mechanism to take this non-live preparation of CSPP to field. And this is where ATSBs, or attractive toxic sugar baits, come into play. So ATSBs take advantage of mosquitoes' biology. So mosquitoes, in addition to the blood, they need to feed on sugar solutions. And both male and female mosquitoes feed on sugar solutions. So this attractive toxic sugar bait is comprised of a sugar solution which is highly attractant to mosquitoes. They come and feed on these sugar baits, which is laced with insecticides, and mosquitoes die. So for this project, we are collaborating with a company from Israel, West Ham Corporation. They have developed an attractive sugar bait, which is attractive to many species of Anopheles mosquitoes. If you see here in the picture, this small little pouch is the ATSB. It contains a membrane, and inside that membrane is an attractive sugar bait. So the mosquitoes come and feed on this membrane, and then they die. The good thing about this is it's very easy to deploy. You can just hang it outside the house, and it lasts the whole transmission season. Another feature of this ATSB is that it's made up of a membrane, which is only permeable to mosquitoes. So other insects, even though they're attracted to this ATSB, they're unable to feed on this, which makes it extremely friendly for the environment. So our next step was to see if our CSPP preparation is compatible with this ATSB. So our experiment is very simple. We grow CSPP in the lab to very high optical density, and then we create this non-live dry powder of CSPP. So at this stage, there are no live bacterial cells. All the bacteria are dead. And we mix this with the attractive sugar bait, which West Ham provided us. And then we carry our mosquito mortality assays. So our first step was to, of course, test if this preparation is effective against lab mosquitoes. So we tested Anopheles gambi Keeley strain from our lab here, and then two other strains from Burkina Faso. One was Anopheles kalutzii, and the other was Anopheles gambi Kisumu strain. As you can see here in the graph, the green line shows mosquito survival when they were fed only with the West Ham bait. And then the pink line shows survival when we added our CSPP preparation to the bait. As you can see, all the lab strains that we tested are highly susceptible to our CSPP ATSB. Now, once we confirmed this in the lab, we wanted to see if this works in the field strains. So for this, our collaborators from Burkina Faso went out and collected Anopheles larvae, 2 sites in Somoso and Poo Valley. They brought back these larvae to the lab, and these larvae were reared to adult stages, and then they were fed with the same preparations, either West Ham bait alone or the bait mixed with our CSPP preparation. And just like the lab strains, our field strains of mosquitoes were also highly susceptible to our CSPP preparation. So in order to put this in the field, it needs to be stable for a really long time. So next test was to see if our preparation is stable. So there is a quick test that you can do. You can, of course, put this on the shelf for a couple of years, or there is something called an accelerated shelf life test, where you incubate your preparation at 52 degrees for 2 weeks, which is approximately equal to 1 year at room temperature. I don't know who calculated this, but this is how it's done. And we also incubated this at 70 degrees at a much harsher conditions. And we saw that our preparation, even after incubation at 52 and 70 for 2 weeks, is still effective, and it does not lose its mosquitocidal activity. So far we confirmed that our ATSB is effective against lab strains, field-derived strains, and it's highly stable. So our next step was to take this to the semi-field study. So for our first experiment, we did not actually have access to the field, to the pouches that I showed you in the first image. So we created some pouches in the lab with Ziploc bags and trash bags, and they were sent to Burkina Faso. And the image that you see is the semi-field facility in Burkina Faso. It contains 4 replica huts, and the whole facility is covered with a fine mesh. And these pouches were placed inside this facility. We had a control pouch that contained only West Ham bait, and then different concentrations of our CSPP. 400 mosquitoes, 200 male and female mosquitoes were released in these huts. And this CSPP ATSB contained a fluorescent marker dye, so we could see if mosquitoes were actually feeding on this. So next day, our volunteers went inside the facility and collected the mosquitoes that were still alive. These mosquitoes were then brought back to the lab. They were observed under the microscope to see if these mosquitoes have actually fed on the ATSB, and then they were sorted as fed, unfed, male, and females. So these are the results from our first semi-field trial. As you can see here, all the females and the males that were fed on CSPP ATSB at different concentrations died in the lab over the days. But the control ATSB that only contained West Ham, those mosquitoes survived. And the unfed males and unfed females also survived in the lab. This showed that our semi-field trial worked successfully, and our CSPP ATSB can perform well in the semi-field conditions. So all these data that I showed to you so far, we are growing CSPP in the lab in our culture flask. But if you want to take it in the field, you need a larger scale production of CSPP. So I'm not going to go into the details of this, but we have successfully established protocols to produce CSPP preparation in a large scale facility. So currently we are carrying out some assays with this new preparation. And actually we found out that our new large scale preparation is even more efficient than our lab preparation in killing mosquitoes. So in summary, I would say that our CSPP ATSB is effective against lab and field strain of Anopheles mosquitoes that we tested. And our shelf life assay shows that the CSPP ATSB is very stable, and it performed exceptionally well in semi-field settings in Burkina Faso. So currently, we are going to do another trial in Burkina Faso with the actual pouches. So this time we actually have access to those bait stations. So we are going to load this ATSB in the bait stations, and hopefully we'll see a similar results like last time. We are also doing some non-target toxicity assays in collaboration with USDA. So they are testing this ATSB with honeybees to see if it does kill honeybees. And we're also working on finding out what is the active ingredient in this preparation, because right now it's only a non-live bacterial powder. It contains a lot of debris and everything. So we are trying to find out what is the active ingredient that's killing the mosquitoes. And with this, I would like to acknowledge especially the Burkina Faso team, Abdullah Diabate, Sare, and Etienne, who helped setting up all these semi-field assays. And without their collaboration, this project wouldn't be possible. I also want to thank Mira from our lab. She's been extremely helpful in setting up all those bioassays, and Yume, and of course, Chris from the JHMRI Insectary Core for providing mosquitoes. And we are also thankful to Ashik from Penn State University, who's helping us with the production of large-scale CSPP powder. And I also want to thank all of you for coming to the talk today, and I'll be happy to answer any questions.
Have you ever tried to see the weed CSPP, but without CSPP have attraction? So the weed the CSPP has some repelling, like a volatile, they're smelling.
Yeah, we tried that in the lab, but the problem is the cage is so small that you cannot really get very effective data. So maybe like with the Y tube or something, we can do that, but we haven't tested that yet on like a large scale.
I think it's the long line of a tube that one end would net with the CSPP versus without CSPP.
Yeah.
Same bait. You can test how many
mosquitoes are attracted. Yeah, that's a good idea. We can do that. Thank you.
What are the ecological downsides of this research to mosquitoes in the area and the toxicological effects in humans?
So this ATSB, like I said, it's designed in such a way that it only allows mosquitoes to feed on the bait. So other non-target insects are not affected. And as far as humans are concerned, we are not spraying this to a broader area. This is contained in a pouch. So I think it's more safer, I guess, than than spraying the insecticide everywhere. Thank you.
Right. This is very interesting. I'm just wondering if have you guys tried just by chance with small mammals? If this is eaten by a mice, it would be nice.
No, we haven't tried.
But if it's eaten by a child,
yeah, we haven't tried with the small mammals yet. Yeah, thank you. Yes.
Has Chromobacterium been described as a human pathogen ever?
I don't know about this particular strain, but like many other bacteria, it has been reported as an opportunistic pathogen for people who are, immune compromised. But our preparation doesn't contain live bacteria, so we don't have to worry about it being infecting someone. Yeah.
Do you have any information on the mechanism of action of the insecticide? And are you taking any steps to get to know?
Yeah, we are actually working on identifying the active ingredients in the preparation. So we have sent some samples for metabolomics and proteomics. So I think in the next coming months, we'll have some idea about what the active ingredient is. Yes.
So it looked like in your lab studies, the preparation was able to kill the mosquitoes for 2 or 3 days. In your semi-field studies, it took like a little bit longer for them to die. Do you have any hypotheses as to why maybe those mosquitoes took a little bit longer to die?
Yes. So one hypothesis we have is we only collected live mosquitoes, right? The next day. We couldn't actually, it's very difficult to actually go and count the dead mosquitoes in the semi-field facility. So our guess is the mosquitoes that had enough, they just died and we couldn't catch them. And the mosquitoes that slowly died had ingested lower amounts of CSPP. And that's why they died slowly. Yeah, that's the current hypothesis.
What is the average time for a mosquito to die? Do you have any information on how long it takes for it to take effect?
I don't know exactly in hours, but we measure mortality the next day and we see almost 60 to 70% mosquito mortality the next day we measure.
Yes, thank you. Is there any understanding of what proportion of female anophelines in a real world environment where there are humans and animals will actually feed on the sugar bait? Is it because if it's only a small proportion, obviously it's going to have much less of an impact.
Yeah. I'm not sure if I have the exact answer to that, how many times they feed on this bait station. But if there is an interesting information on the West Ham website, I don't know if it's published, but they have done some modeling, and according to their modeling, if only 2 to 3% of mosquitoes feed on this, it should have enough impact to reduce the malaria burden, approximately 20 to 30% in the area. I'm sure they have done some modeling, but I am not entirely sure about that.
I'm going to take one more question.
Do these also have effects on the parasites?
We haven't tested the non-live preparation, but the live bacteria does block parasite infection in the mosquito.
Because depending on the level of feeding or the amount of cells that the mosquito takes in, even if you get delayed death or some mosquitoes that they don't die, if they have an impact on the parasite, that can also have an additional impact.
Yeah, that's a very good point. I think we should test that in the lab. Thank you.